everybody, it's Miss Peachy again from WCA Biology, and today we are going to be doing Unit 8, Lesson 5, Genetic Mutations Produce Variation. So this lesson is a lot of it's review, I think, from previous lessons. And we do introduce another term that you haven't seen before. So our keywords here are inheritable genetic variation, mutations, homologous chromosome, zygotes, population bottleneck. So let's kind of go through those. We've talked a lot about inheritable genetic variation. So these are variations in a population that can be passed on from parent to offspring. And as you know, that means that those traits need to be present, or those variations need to be present in the egg or sperm in order to be passed on. They just can't be passed on any other way. These inherited genetic variations come into existence often through mutations. So, excuse me, where do those mutations happen? Well, they can happen in multiple places. They can happen as a result of changes in meiosis, right? Or they can happen due to environmental effects as well. So first of all, homologous chromosomes. What are they? Again, so homologous chromosomes are, remember you have two copies of each chromosome. One comes from mom and one comes from your dad. The two copies are homologous to each other. That one's from mom, that one's from dad. They have the same genes on them, but there's different alleles. So mom might give you the blue eye allele, whereas dad might give you the, I don't have brown as a color, so I'm going to go with black, the brown-eyed allele, okay, for eye color. They both, on the same chromosome, you have the same genes, but there's variations in them because one comes from one parent, one comes from the other. These two are homologous to each other. They have the same genes, but they have slight differences based on the characteristics you're inheriting. During meiosis, or just prior to meiosis, each chromosome makes a copy of itself. And each of those, you've got two of each now, right? These two orange guys are sisters. So those are our sister chromatids. And we see sister chromatids only during replication. Replication to form eggs and sperm is called meiosis. During meiosis, you have things that can happen that can lead to variation. You can have, let's see, I'm going to go down to the bottom here because I think they talk about it down here. You can have crossing over, which happens during prophase 1, the first part of meiosis, meiosis 1. And that's where these guys become entangled with each other and start swapping a little bit of information. So a little red ends up over here, a little orange ends up over here, whoops, over here. They're still the same genes, they're just the alleles kind of get swapped a little bit because they get messed up. That's crossing over. You also have independent assortment. As these move to different cells during meiosis 1, you're going to have the splitting up that happens. So, half move to one new cell, half move to another new cell, and eventually during meiosis 2, those sister chromatids separate. But because there's 23 combinations, some of the red go one way, some of the yellow go the other way, and we have that happening 23 times. You have a random number of combinations that can occur. That's called independent assortment, right? I know I mentioned to you in a previous slide, but with 23 pairs of chromosomes, there's like over 8 million combinations that you can make with your eggs and over 8 million combinations you have in your sperm because of how they independently assort. So that leads to variation too. The other thing that can happen is something called disjunction, which we haven't talked about before. So this is kind of a new term here. So this is where the chromosomes, they don't separate properly. So you may end up with a different number of chromosomes or fragments of chromosomes sometimes. Sometimes they break apart. So those are chromosomal issues that can happen. Maybe you'll get 3 copies of one instead of 2, right? Or maybe you end up with or I should say maybe you get like an extra copy. So then after fertilization, you have 3 instead of 2. Or maybe one of them breaks apart and you end up with just a part of a chromosome. Those can often lead to more catastrophic results than if they separated properly and you just had like other mutations. The other things you end up happening to cause mutations are sometimes errors in the whole process of replication. So I love this down here. Where is it? Right here. You end up with mutations in the DNA that can be, they can be a bunch of different things. You could have a substitution, maybe an extra nitrogenous base gets plopped in there, right? Or I'm sorry, that's where one was substituted for another. It's supposed to be an adenine, and instead they put a thymine in place. So it was just the incorrect, a spelling error, if you will. Or you can actually squeeze in an extra one or delete one. And these two here can result in frame shift mutations. So DNA is transcribed into messenger RNA, which is then read and translated into an amino acid. And every messenger RNA strand is read 3 bases at a time, right? So if the messenger RNA strand is supposed to be U, G, G, C, A, G, and it's going to be read like that, okay, that's one amino acid, that's another amino acid. If we add one in the middle, U, G, 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 C, A, G, suddenly it's going to be read like this. Oops. There's 2 totally different amino acids, right? This was supposed to be one of our amino acids, and now we get that instead. This was supposed to translate into our second amino acid, and now we're going to get this instead. So a substitution, or not a substitution, an insertion, that's an insertion error, can do that. Also, a deletion error could do that, right? So instead, if I deleted, say, one of these letters here, right here, let's say my next letter here was U, now it's going to be read wrong. It was supposed to be CAG, now it's AGU. So again, both of these guys here can result in frame shift mutations. And that just messes up your entire messenger RNA, all of your amino acids after that point. And obviously that, I mean, the proteins aren't going to be translated or formed correctly at all. It's really pretty bad error. So again, that's another mutation that can result. And when we look at down here, we see, like I said, I like this because it says if you have a mutation in the DNA, so some sort of error in the DNA, whether it's an insertion, a deletion, or a substitution, that will ultimately change your RNA, which will ultimately lead to changes in the amino acids and proteins, and it ends up changing your traits. So it's another way to introduce genetic variation. Where, oops, go down a little further. Where do these, why does this happen? Well, it can happen due to problems in the environment, for example. Lifestyle choices, diet, stress, and exercise, as well as things you can't control as much, pollutants and exposures to toxins and radiation. So these all things can lead to mutations. And then, let's see here. Other thing, oh, I wanted to mention, let's see if I can get down to it. Okay, something called bottleneck or population bottleneck. So here's a change to genetic variation in and of itself. So mutations cause more variation. A lot of times those mutations are bad and they actually cause variation that's not something that we want or keep around in a population. A lot of times they're lethal. They don't really result in any changes, but they do occasionally result to beneficial changes in a population, which is, a good thing. What results in a reduction of genetic variation One of those things is called a bottleneck, a population bottleneck. And basically what happens with a population bottleneck is that the the population is drastically reduced in size because of some external factor that has nothing to do with any of the characteristics. Um, we look at this, we see this often with human caused problems. We completely destroy a habitat that destroys a lot of the organisms in that habitat and leaves just a few behind. And what happens in this situation is that you lose variation, you lose genetic variation because you've destroyed many of the organisms in that habitat. The traits that those organisms had are gone and cannot be passed on to a next generation. If the organisms that were destroyed were the sole owners of those traits, then those traits are gone forever from the population, right? So that's what's known as a population bottleneck. A population is dramatically reduced in size, and it actually results in loss of genetic variation because the organisms that were killed were the only ones that had the traits, and you've removed those traits from the gene pool forever. That's what's known as a population bottleneck. Okay. All right, so that is everything from this lesson. If you have questions, let me know. Otherwise, ladies and gentlemen, I will see you the next one.